we will transfer presenting rights to Jonny here. While we're doing that in very real time, I'll note that Jonny was a graduate student of Dr. Tom Gill. Since graduating, he's been working as a food scientist for Perennia Food and Agriculture, which is Nova Scotia's provincial food development agency. He gets to apply every day his passion for food and work with seafood companies as they overcome various technical and process hurdles to innovate and meet changing consumer demands. On a more personal note, because I learned this about Jonny during our prep call, Jonny has a dog, a 7-month-old Labradoodle, and I'm waiting for my own dog to start deciding that he has to be part of this webinar too. But with that, Jonny, how about if you kind of tell us, dare I say, the next phase in the research?
Sure. Can you see my slides okay?
Yes, got your slides and you're coming through great. Thank you.
Perfect. So thank you, Gwendolyn, and thank you everyone for being here and letting me close off this series today. Yeah, so I'm going to talk about identifying low molecular weight peptides, which is the theme of the day. And Tom already discussed briefly about the salmon protein hydrolysate and its anti-diabetic activity, and Dr. Aluko kind of referred to some of the other bioactivities that that hydrolysate possesses. And so, ultimately, if we want to understand, the mechanisms here, or eventually lead to IP, we want to identify what those peptides are that are mediating that activity. So I'm going to walk through today the process of how we identified some of those bioactive peptide sequences from that salmon protein hydrolysate. I have no affiliations or conflicts of interest that I need to disclose. And just as a brief outline of what I'm going to talk about, I'll just give another brief overview of food protein hydrolysates, why they're important. And then I'll kind of talk about the approach to how we go about identifying what the sequences are in hydrolysates, and in particular, how that is a particular challenge for those of low molecular weight. And that's, again, consistent with a less than 1,000 Dalton definition. And then I'll review some of the data that I collected through my PhD project. And as Tom mentioned, we just went through a process to patent this information, which concluded this summer. So most of this, or all of this research, is really unpublished so far. And then we'll just summarize what we learned. So as Tom mentioned, these food protein hydrolysates are really any hydrolysate from dietary proteins. And as he also mentioned, one of the real drivers for producing these types of products is the reality that producers are looking to maximize the value that they can gain from the products they produce. So as opposed to diverting things to waste or other low value streams, certainly trying to develop either pharmaceuticals or other functional bioactive compounds that can be sold is a better option for everybody. And so some of those markets for hydrolysates are pharmaceuticals, like we've seen. A lot of those bioactivities are can be exploited and created into some type of drug. Food industries certainly are seeing a lot of applications for hydrolysates, and in particular, I think the marine collagen hydrolysate is a very popular product now in the market, and it's being added to food, it's being added to cosmetics, and ultimately it's a great use for fish skin and fish bone. And of course, biotechnology, there's other applications for using hydrolysates to stimulate growth in many ways. And ultimately, with all of these protein hydrolysates, it's the bioactive peptides within them which are considered to be the functional drivers of these mixtures. And generally speaking, we're talking about 2 to 20 amino acid length peptides. Anything larger, you might call it a peptide hormone, but really, the exercise to try to identify it is quite similar. And when we're looking specifically at low molecular weight hydrolysates, that can include everything from free amino acids, dipeptides, tripeptides, and so and everything greater than that. So there's a lot of complexity to what that composition might be. And again, when you're trying to produce this, say, at a commercial level, Tom showed that 1,000-gallon tank. When you're adding 300 kilograms of salmon burger and hydrolyzing that with trypsin, pepsin, and chymotrypsin, you are generating thousands and thousands and thousands of unique components. And so how we go about identifying them really needs to try to capture all of that variety. So Dr. Oluko also touched on the general strategy to how we go about identifying peptides from bioactive peptides from hydrolysates, and that's typically with a fractionation procedure. And he summarized it perfectly by saying, this serves to purify the peptides to make them less complex and also to concentrate the bioactivity, ultimately making those fractions more easy to study than the whole hydrolysate itself. And there's a growing a growing opportunity with bioinformatic tools to do or mimic a lot of what we're doing experimentally with computational means. So with the availability of primary sequence information in online databases, we can now simulate digestions and predict peptides and predict what some of those functions might be with other previously known information. But we have none of that for this salmon protein hydrolysate. So ultimately, to be able to capture the complexity of these types of samples, this field of study is sometimes referred to as peptidomics, which is like proteomics, trying to quantify and identify the components of these unknown peptide mixtures. And really the only molecular technique that's capable of detecting this range of composition in these samples is liquid chromatography tandem mass spectrometry. And while that technique is effective at detecting the composition of the sample, it's really how that process is coupled with a database searching process that is necessary to assign sequence information to the compositional information that we detect through LCMS. So I'm just going to kind of give you an idea of how that process was originally designed for proteomics and how it's being applied to peptidomics. So this is a figure here that really summarizes the technological status of each stage of the database searching workflow. So let's say you're starting with a protein extract with a number of unknown proteins at various concentrations, and you want to understand what those proteins are and how much of them are there. Starts with hydrolyzing that protein sample, and then you separate and detect it by LCMS, and then there is a software-assisted bioinformatic approach which looks at protein database information, and then you compare your experimental data to some of that database info. And so that whole process is very well established for protein identification. But as you try to apply that to protein hydrolysates and samples of much smaller compounds with a much higher degree of complexity, these steps aren't quite as transferable. And ultimately, in the end, you have a much higher rate of false positives and false negatives. But nonetheless, this approach is really the only high throughput process that exists at this time to be able to scan these hydrolysates and try to assign sequences onto them. So given these limitations, we tried to make improvements to this process to be able to increase our confidence ultimately in the peptide sequences we identified following this procedure. So I'm just going to give you a little bit more detail about how that works for proteins in particular. And so let's say we have a sample, it's a pure actin protein from, let's call it Atlantic salmon or Salmon solar. And so the first step in that process was hydrolysis. And typically for proteomics, that's done with trypsin. So you have your protein and you treat it with trypsin, and each one of the lysine and arginine residues in that protein are effectively cleaved, and you now have a peptide sample. You take that sample and you would inject it into the mass spectrometer. It would first get separated by typically reverse phase liquid chromatography, and then individually those component peptides would be injected into the mass spec. In the first stage of mass spec, each one of those individual peptides would be detected and a mass would be recorded. And then further, each one of those individual peptides is transferred into what's called a collision cell, where it gets treated with a burst of energy and non-specifically fragmented into, a random collection of ions. And it's those ions that get detected one further time, and that represents what's called an MSMS mass spectra, which is there on the bottom. And so that is ultimately almost like a peptide fingerprint of what that detected ion might be. And that is all really that the LCMS does. You get masses for your ionized peptides, but no other information. And so that's where we rely on the bioinformatics to interpret what those peptide sequences might be. And it all starts with having a known proteome for the species you're working with. So the software, you input what species you're working with, in this case, again, Atlantic salmon, and then you'll tell it how you've processed the sample originally. So in this case, we've cleaved it with trypsin. And so that whole database of protein sequences will then be shifted into a peptide database of tryptic peptides from that species. And then with those tryptic peptides, the software essentially creates a theoretical MS mass spectra. And it's through the comparison of your experimental data through the LCMS and this theoretical spectrum and the similarity between them, that is how an identification event occurs. So when there is a certain degree of confidence between that theoretical spectra, then a presumptive peptide identification has taken place. And of course, this is raw data, and that needs to be further validated using various scoring. But ultimately, those peptides which get identified are then used to identify what those original proteins might be. So when there are a sufficient number of peptides identified from these original proteins and a sufficient coverage of that initial protein, that whole protein was identified, that ultimately is how a peptide identification and protein identification is completed. So as this is applied to protein hydrolysates, you're really eliminating that first step where you're treating it with an enzyme, because your process of creating hydrolysate serves that same function. And likewise, this last step of where you're relating the identified peptides to the progenitor proteins, that's not really needed for identifying the peptide sequences in a hydrolysate. So briefly, the salmon protein hydrolysate that we were working with, Tom mentioned, started with a deboned salmon mince that was solubilized in 1 molar sodium hydroxide. Through pH shift, we isolated an isoelectric precipitate that was then hydrolyzed with pepsin, trypsin, and chymotrypsin, and then we concentrated that to the less than 1,000 Dalton molecular weight cutoff. Really, this range will capture, mostly less than 6 amino acids, but if we're talking peptides, 2 to 6 amino acids. And again, for the purposes of trying to identify what the bioactive peptides are, we're looking at this from the lens of the glucose uptake stimulation activity that we had, Tom had mentioned. So my goal here was trying to identify what the peptides were, which were contributing to this glucose uptake stimulation. And it's important to consider that, there were no other, in the literature, there were no other peptides which had claimed to have the same effect that we had observed in our bioassays. There were some other peptides that did show to have some glucose uptake stimulation, but whereas those were shown to be active at 1 milligram per mil, our peptides were effective at a nanogram per mil concentration. So far, far more potent. And again, as Dr. Aluko mentioned, fractionation, that's really the strategy here, to try to concentrate the activity and get further purification of those fractions to make the identification process a little bit simpler. And, again, there are potentially thousands of peptides that can be generated with the specificity of these enzymes that we've used. So this endeavor is really trying to identify a needle in a haystack type challenge. So the first process we followed, and unlike Dr. Aluko, who fractionated by reverse phase chromatography, my first attempt was using a strong anion exchange chromatography. And the reason why I did that was because I wanted to understand if the charge on the peptide had any influence over that glucose uptake stimulation. And what we found was that both of these two fractions, which I had generated, had a stimulating effect on glucose uptake and was also no different from the unfractionated SPF. So, the fractionation ultimately doesn't tell me anything. It doesn't help to give me further purity of the sample or really aid in the identification. But nonetheless, we try to identify the composition of these fractions. And for our first attempt, we were following that original proteomic methodology for database searching. And so when we plot the mass and charge of all of the individually identified peptides, we can see that nothing less than 600 Daltons was identified, which is really the main range of mass that we were hoping to capture and that we thought would be most abundant in our fraction. And we were, I think, quite surprised to see that peptides exceeding 1,000 Daltons here were found in the sample. And even though we didn't identify anything less than 600 Daltons, there's still about 500 peptides here that were identified. So we went back to the drawing board and tried some other fractionation procedures. So this next attempt was using a gel filtration chromatography, and the stationary phase that was used was Biogel P2. It has a 100 to 1800 Dalton molecular weight range. And through the separation, we collected much smaller fractions, 13 of them, excuse me. And 6 of them were found to have some type of biological effect. And actually, for the first time, we found some peptides that had stimulating effect as we have seen before, but also an inhibiting effect on glucose uptake. And this, I think, threw us for a little bit of a whirl, because while we had, initially hypothesized that there would be, a peptide that was contributing to this biological activity, now we're getting a little bit more information that this might be a little bit more of a synergistic effect between multiple peptides. Strong anion exchange was completed a second time. This time was using a much longer column, which allowed us to capture more of that anionic range in the peptide. And again, 3 fractions closer to the end of the elution strategy were identified to have a stimulating effect on glucose uptake. And so this, is maybe suggesting that it's those strongly anionic peptides which are contributing to that biological activity. So using these 9 fractions, we try to study them further and detect what their composition is to give us maybe a better understanding of what it is that's contributing to that bioactivity. And this time around, we try to improve upon the database searching workflow that originally did not capture the full complexity of the low molecular weight protein hydrolysate. So again, here's our actin protein, and this time I've bolded all of the residues, which are the cleavage sites of pepsin, trypsin, and chymotrypsin. And we can see that this is 3 to 4 times more cleavage activity in our original sample, and nonetheless we analyzed that sample by LCMS in the same way. But the challenge with low molecular weight peptides really comes into play with the bioinformatics of this process. So whereas previously the instructions you're providing that software is that you've cleaved your sample with trypsin. Unfortunately, the software for this tool does not allow you to input 3 enzymes. So how we went about doing that was by looking at pepsin hydrolysis alone. We looked at trypsin and chymotrypsin together, but ultimately, those don't capture the full complexity of our sample, and so we adopted what we're calling a no enzyme specification, which takes the full proteome of primary sequence data, and using our 3 amino acid cutoff, which is what the lowest the software allows us to do, we take every possible tripeptide in that protein database that can be created, and we create these theoretical spectra from that. We do that also for every tetrapeptide and every pentapeptide, and in this case of actin, we'll do that for every length of peptide up to 374 amino acids. So the sheer complexity of these databases that you are ultimately comparing your experimental data to just becomes so big and so complex that this is where the source of a lot of your false positives and false negatives are originating from. And so this highlighted section of actin, these peptides that I show here are the outcome of just that small sequence of actin. And we're looking at 3 amino acids, dipeptides, many tripeptides and tetrapeptides, and some peptides that are larger than that. But even with our bioinformatic process, we've already excluded free amino acids. We've already excluded dipeptides because the software can't tolerate it. So if our effort is to try to identify all of the peptides in this solution, there does not appear to be a high throughput, one-size-fits-all solution that will tell me the complete composition of my low molecular weight hydrolysate. And on top of that, of those peptides that are there, there's still the uncertainty of how accurate those are because of the sheer size of the proteomes that we're dealing with. So again, it's the high throughput which is really needed to be able to tolerate these samples. And so this table that I'm showing here is really the output of the database searching that is completed. And in total, from those nine fractions, there were about 1,000 potential peptides that were identified. And this is raw data, and there's scoring here and other parameters to filter the data. And what is recommended if this had been a proteomic procedure is to filter out any peptides that have scores that are, say, less than 9. And so if we're following on the table on the right for good quality data, we would exclude any identification with a score of less than 9 and a % SPI that's less than 60. And that is just the degree of similarity between your experimental and theoretical spectra. And when we apply those validation criteria that are recommended, we then end up with 71 peptide identifications. And so that excludes over 90% of the peptides that are possibly identified. And it also had a bias based on the score to peptides that were larger than, let's say, 4 or 5 amino acids in length, which we had thought exceeded that target range of the peptides that we were interested in. And if we also look at the proteins which were considered to be the progenitor sequences of the peptides, we're looking at here hypothetical protein AWJ, a hemoglobin subunit, a copper transporting ATPase. These seem to be not what we would expect to be the primary proteins contributing to bioactive peptides from a whole muscle extract from Atlantic salmon. The myofibrillar proteins, myosin and actin, we thought would be contributing most of the peptides that we were seeing in this mixture. So because the sources seemed a little bit obscure, we wanted to improve upon that to some degree. And the proteome that we're working with is derived from this website, which Uniprot. It's this is the source of online primary sequence data, and we're using all 86,000 possible primary proteins that are held within this database. And so again, when we're considering these 86,000 possible proteins and the approach to how we generated a peptide database from these protein sequences, we have potentially hundreds of thousands to millions of individual peptides that we're comparing our experimental data to. So if we can improve upon how we do that, then I think we'll have much higher confidence in the identification process. And so with the with these bioinformatics software that are used to operate the LCMS, they offer some additional tools to help you study your data. So one of those tools is called a molecular feature extraction. And ultimately what that is it's an algorithm which deconvolutes your LCMS data and gives you a list of very high likelihood compounds or features within your hydrolysate, which are considered to be real compounds in your sample. So for every fraction that we generated, we could apply this algorithm to, and we could understand, we get a retention time and ion mass for every presumptive real compound or peptide in that fraction. So if we have this list of ion masses, and we also have these database search results, I propose to do a little cross-reference of those two sources to improve how we validate those database search results. And so when we look at the individual fractions, we can see that without any validation, certainly there's a high number of peptides identified in each. Compared to the recommended procedure, which the recommended validation process, where you have a high score requirement, that excludes, again, about 90% of the sequences. But when we integrate that, the molecular feature extraction into the validation, which is what we're calling the modified process, we do get about a 4-fold increase in the peptides identified in each fraction. And so if we also compare what the molecular weight range is, or the molecular weight distribution of each of those fractions, depending on how we validate that data, we can see that that recommended procedure actually prefers a higher molecular weight range than the raw data from database searching, as well as from the output of that molecular feature extraction. So the modified validation process is more in line with some of the raw data and certainly captures the low molecular weight range from 200 to up to 700 Daltons. So now that we have a little bit more confidence in some of the peptide identifications from database searching, we want to effectively analyze what those peptides are a little bit better. So depending on the original chromatography, so the gel filtration fractionation, the molecular weight distribution of those fractions were actually all found to be quite similar, ranging from 200 to 600 Daltons, whereas the fractions from strong anion exchange actually were more so concentrated between 400 and 800 Daltons. And while we would have expected for the fractions from gel filtration to have progressively smaller peptides in them, this was not observed. And so when we look at the peptide sequences within them, which this shows as predominantly tripeptides, those fractions that had glucose uptake stimulating activity showed a fairly similar composition. So most frequently, there was an isoleucine residue on the N-terminal and the C-terminal. But in the fraction that eluted earlier from the column, a higher polarity residue was found at the middle position, whereas a more neutral amino acid was found at the center position in a fraction eluting later. And in an even later eluting fraction, we're now seeing, again, a sample that was predominantly tripeptides in composition, but those peptides now more frequently possess tyrosine residue on the C-terminal. And so while we did this chromatography to separate peptides by size, what we really found was that the tripeptides were separating on the basis of hydrophobicity. And so although we're not able to make any conclusions about peptide size as a result of that fractionation, we do get some ideas about maybe there is a sequence fact, there's some type of compositional prediction on what that activity is based on the sequence. So given that the fraction F6 had an inhibitory effect and had a unique compositional feature with that aromatic amino acid at the C-terminal, we predicted that it could be this unique amino acid primary sequence that may have been influential to modulating glucose uptake. And so some of these sequences were, in fact, ones we chemically synthesized and under purified conditions confirmed whether or not they had glucose uptake stimulating or inhibiting activity. So as I mentioned before, the database searching process did not capture dipeptides, and we were very, very certain there were dipeptides in our sample. And fraction 9 of gel filtration in particular, we found to have an inhibitory effect on glucose uptake, but the database searching process really did not identify peptides that we were confident with. The one sequence that we identified, TWPW, was actually identified in a few fractions, which tells me it's probably contamination. And the feature extraction algorithm did, however, suggest there were 27 compounds in that sample. However, when we look at the total ion chromatogram from mass spec, we can see very clearly that there appears to be just 7 peaks or peptides of interest that would be important for us to further study. And to do that, we can look at those MS spectra, which are the fragments of the initially identified ion, and we can try to decipher manually what that amino acid sequence might be just by looking at those individual fragments. And so for each of these 7 peaks, we were able to assign a peptide sequence of them using this approach. So for peptide, for ion B, it was a dipeptide with sequence of alanine and tryptophan. For the next ion, it was valine and tryptophan, and so on. But nonetheless, again, we're seeing here, again, a sequence similarity to that previous fraction that also had an inhibitory effect. So we see the terminal aromatic amino acid, I think, very much might be influential to the bioactivity that we're seeing in the sample. With these known dipeptides, we can actually refer to other bioactive peptide databases to see whether they possess any other types of activity. And so when we do that, we find that many of those sequences have similar functions, including angiotensin 1 converting enzyme inhibition that Dr. Oluko had identified. And just as he showed that it was the fractions that allude further on in the reverse phase chromatography, which suggests a higher hydrophobicity. Here we are showing again peptides with a hydrophobic amino acid, and it's very likely that some of these peptides could have been contributing to the activity that they had observed. So the challenge with the very complex proteome that we were originally dealing with was one that I wanted to overcome. And so I asked myself the question, which is, we're producing this protein isoelectric precipitate. Can we resolve that precipitate and get a better understanding of what it is the main proteins are that we are hydrolyzing with our sample? So in my attempt to do that, in column 2 here, we can see the result, which is absolutely nothing. And as we learned, we found that the concentration of sodium hydroxide, as well as the time of incubation in sodium hydroxide, seem to be a factor contributing to the poor resolution of the proteins which we precipitate. It was only when we instead of solubilizing in 1 molar sodium hydroxide, but instead used 0.1 molar sodium hydroxide, were we able to get much better resolution of what those proteins were. And so just like the proteomic methodology, we wanted to try to identify what those proteins are. So we can cut those bands, those individual gel bands out of the gel. We can, we would destain them, you reduce and alkylate, and then you would hydrolyze them with trypsin, and then you take that tryptic hydrolysate out of each band and try to determine what those protein sequences are, or those individual protein identities. And I got the idea, if we're trying to do in-gel digestions for protein identification, we now have these purified proteins that are what we're considering to be the sources of bioactive peptides in the protein hydrolysate. can we, instead of just tryptically hydrolyzing them, but replicate the whole process that we've used to produce the salmon protein hydrolysate in this gel? And so as an alternative, we excise those gels, we hydrolyze them with pepsin, trypsin, chymotrypsin, and then we try to filter out or concentrate the low molecular weight fraction of that result. And then trying to validate that bioactivity, we found that those proteins, which, as it turns out, were the myosin heavy chain, actin, tropomyosin. So certainly those myofibrillar proteins that we were after, but also many of those enzymes involved in glycolysis and energy utilization. And nonetheless, the result of that was even more inhibition of glucose uptake and not quite stimulation, which we were looking for. So while we were trying to identify maybe, which is the protein that was contributing to the bioactive peptides that we were trying to identify, we were really unsuccessful here, and the sheer inhibition across the board, we kind of, lost confidence and, moved on to something else. But I thought it was a neat process in order to, go about the identification process slightly differently than it's typically done. And, you can gain a lot of other potential opportunities by purifying the individual proteins to begin with. And so these protein identifications that we achieved by SDS page or electrophoresis actually were quite similar to the same proteins that we had identified initially using our proteomic approach to database searching. And so this gives me much greater confidence in what are the proteins that are actually contributing to the bioactive peptides having an effect in the STF. And unlike the previous peptidomic methodology with database searching, if we exclude all of those sequences instead, then I think we can certainly improve the confidence we have in the identification that we assign to those peptides. And so the last strategy that I tried was really trying to bring all of that together, which was, if we can apply database searching and we get no result, then how can we get more information in a more higher throughput process than, say, just de novo sequencing, which we have done. So using those 9 proteins that were identified, we then perform what's called an in silico digestion. So we're trying to simulate the hydrolysis process of those proteins, and we're using the predicted peptides that come out of that as a possible validation of the de novo sequencing process or either the database searching process. And in addition to the specific hydrolysis, which is predicted by the enzymes, we're also looking at more non-specific hydrolysis. So if we have our feature extraction with known peptide masses, we can mine effectively the primary sequences of those 9 proteins for which are possible sequences that can be that match those masses which we have identified experimentally in our sample. And so for this inhibitory fraction F9 again, we through de novo sequencing identified a number of dipeptides, and certainly we determined that with the specificity of the enzymes we used on those same proteins which we also identified, those peptides are indeed predicted and should be in our sample, and that helps us give us more confidence that these peptides are indeed real and were identified. And so that is a process that is slightly more higher throughput than a manual process alone. So in conclusion, pretty quickly, the validation of database searching as it applies to low molecular weight peptides is just very, very challenging and is not a standardized process established yet. So we need to look at other ways to try to give us better confidence in that identification process. And given the complexity of the hydrolysates with, single amino acids, dipeptides, and tripeptides, there's a real need for a one-size-fits-all solution to do this with high throughput. Otherwise, we are, combing through the data and applying multiple techniques to try to understand the full complexity of those samples, and therefore what could be truly contributing to the bioactivity that we observe. And in our particular application, it certainly appeared that it was not just one peptide responsible for any of these activities, but a whole collection of peptides acting synergistically with each other, which are contributing to this effect. So that just is another rabbit hole to go down, but that's for somebody else. Thanks, that's everything I got.
Great, thanks, Jonny. Appreciate your walking us through all of that work, and definitely is a bit frustrating when databases don't quite work how you think they're going to work, and it is inevitably the graduate student who finds that out. So a couple of us may have been, so but appreciate it. I was wondering if you could expand a little bit more on your comment that it's not just one single peptide, it's not just, say these 3 amino acids, but rather it's that synergy component, right? More, dare I say, just talk about what your perspective is on that.
So ultimately, this whole process is identifying what are presumably candidate peptides for that have this biological effect. And if you want to confirm that process involves synthesizing that peptide and testing it in a purified form. So we did that, and the outcome of, let's say, 3 peptides that we tested individually was not the same outcome that we found when those 3 peptides were tested in combination. So ultimately, there is some type of competition or interaction that's happening that, one peptide at 1 nanogram per mil does not quite give you the same result as that peptide at 1 nanogram per mil in the presence of these 2 other peptides at the same concentration.
Got it. Thanks for clarifying that. Want to be very respectful of everyone's time, so I'll ask you this question, and it's loaded. Okay, is having recently finished your PhD, etc., what advice would you offer to those who are still in the midst of that process?
Well, I've always kind of, followed my passions and my interests. And, I originally started with, a biochemistry and physiology degree, but I really liked food and food science. And so that's kind of what led me to working with Dr. Gill. And I've always kind of been fortunate enough to, pursue the things that I'm interested in. And that's always given me, helped me sleep well at night, knowing that I'm enjoying what I'm doing and I'm not, I'm trying to work for myself and not somebody else.
Got it. Very cool, and definitely very, very sage. It helps if you have a genuine interest in your research area. So appreciate your joining us, Dr. Rolin.
Thank you.
With that, want to thank all of you for joining us today. IFINS is all about providing connections, collaborations, and catalyzing science that matters. If you have questions, please visit our website at ifins.org. Follow us on Twitter, and of course, follow us on LinkedIn. In fact, LinkedIn is the best platform to stay abreast of webinars that we offer, RFPs that we issue, and other activities that we support as we endeavor to ensure that science matters in decision making. I'd be remiss if I didn't highlight our upcoming Science Innovation Showcase next week, where we're going to hear from a variety of presentations from government, academia, and industry, both small guys in industry and up and coming emerging brands, where we're going to talk about and hear about some food ingredients. We're going to hear about food safety and more. If you want to register, please go to our website at ifins.org and click on the upcoming events tab, and there you will find our innovation showcase. Lastly, want to thank all of our presenters today, Dr. Gill, Dr. Oluko, and Dr. Rolin, for joining their journey, and it really has been a journey, I think we all heard that, in moving from peptides that might be interesting to peptides that have demonstrable effects. So I want to thank all of you for that. Please note that if you have feedback, I am very happy to take it directly. My email address is shown here on the screen, wendolyn@ifins.org. So have a good day, everyone. Thanks.